Next is keynote address for AIOS Colonel Rangachari Award by Dr. Natasha Pahuja and novel altered critical intracellular signaling events in keratoconus and its therapeutic potential.
Well, it's an absolute honor to be presenting this paper here among such an august audience. What does this paper do is basically we're looking at keratoconus with a slightly different perspective. The mouse is not working. It's not working. Could you go next for me there? Okay, perfect, thank you. So this is the paper that had won the Colonel Rangachari Award last year. And what are we exactly doing is looking at keratoconus with a slightly different perspective. So next. What is the profound thing when we look at this? When we look at this, we feel that the housekeeping mechanism or the cleaning mechanism of the eye is not in place. There's something wrong. This is exactly what happens in the eye, and this process is called the autophagy. What autophagy is whatever is the misfolded proteins or debris, they get collected into a phagophore, and that forms an autophagosome that is included into a vacuole, and all the debris is dissolved or lysed. So this is what is called as an autolysosome. What did we do this? We first created a concept. We had an idea. So the idea is what one actually, fortunately for us, also won the Nobel Prize for the field of physiology in the year 2016. Now, we know that there are certain causative factors for keratoconus, the genetic factors and the environmental factors. There are listed genes and there are few environmental factors the most important of which has been oxidative stress, which has not been touched upon. So we are going to be looking at oxidative stress. Now, why are we looking at oxidative stress? Is because oxidative stress and autophagy are interrelated. That means whenever there's oxidative stress, there's increased reactive oxygen species, hence the inflammation. This causes the oxidative stress and reactive oxygen species. Now, how do we create this experiment? Before going on to the patients, we first tested this in the cell culture lines. Now, we wanted to study the first part of it, that does autophagy or altered autophagy or oxidative stress has got anything to do with the pathophysiology of keratoconus. So in our materials and methods, we formed the human corneal epithelial cell lines, we cultured them, and we induced oxidative stress by inducing hyperoxia. What we did further was to prove this concept. So this is what happens in the lab. We cultured these human epithelial corneal cell lines. We did a quantitative PCR to look at for the inflammatory markers as well as the autophagy or markers. The Western blot analysis further to confirm the autophagy markers and immunofluorescence. And what we found was that at day 7, there was increased oxidative stress in the epithelial cell lines. But what is important is that the autophagy markers are significantly elevated in the patients who in the epithelial cell lines which had increased oxidative stress. This was further validated with the Western blot analysis, which is showing that autophagy is blocked. That means the cleaning mechanism is blocked. This was further seen on immunofluorescence, where the green bodies in the hyperoxic section show that there is increased autophagosomes, which means there is increased oxidative stress. That means there is defective autophagy. Now, we wanted to study this in the patient model. Now, what did we do in the patient model was, we wanted to see the role of autophagy as a pathophysiology in keratoconus. We collected the epithelium of all patients undergoing surface ablation and as controls, and C3R for keratoconus. Now, the epithelium was collected over the cone and the periphery in the same eye of the same patient. This was saved in the biorepository. These epithelium that were collected the patients had very strict inclusion-exclusion criteria. And what did we find in our Western blot analysis was the inflammatory markers and the autophagy markers were significantly elevated in the area of the cone of the same eye of the same patient as compared to the periphery. The periphery was compared as compared to the normals. So what we found was that there is defective autophagy in the ectatic zone of the keratoconic cornea. So what we found was that in the in vitro models as well as the patient models, there is defective autophagy which could be a possible keratoconus pathogenesis. Now what further we did was we wanted to see if we can translate this into the clinic. So we wanted to see what is the pathophysiology of keratoconus, or the pathway of autophagy, and how can this be blocked. So there are few areas where this can be blocked. Rapamycin is one of the targets, but it is not commercially available. The most common commercially available, or at that time an ophthalmic solution, was chloroquine, and we tried to see if it clears off autophagosomes or not. So we tried this again in the human corneal epithelial cell lines. We induced hyperoxia. We saw that there is increase of autophagosomes, there is defective autophagy. Furthermore, we treated them with chloroquine, and we found that the autophagosomes disappear. That means there is optimal autophagy. Now, what do we understand by this? There are environmental factors, there are inflammatory markers, there is increased reactive oxygen species that attack the cells. This is what causes oxidative stress on the cells. Now, this, if the autophagy is altered, that means the cleaning mechanism is altered, there is going to be increased inflammation. This increased inflammatory milieu is going to cause extracellular matrix degradation or collagen degradation. Now we are trying to develop specific drug targets that already decrease the inflammation. Also, they induce autophagy. Now chloroquine happens to be one of them. What we concluded was that autophagy is an important mechanism for cell survival. It causes increased oxidative stress, causes defective autophagy. Now KC epithelium has shown defective autophagy in our study, which happens to be a novel untouched pathway for an alternative pathway for pathology of keratoconus. Also, modulation of autophagy can be one of the therapeutic targets. So what we already knew is that autophagy is known in various diseases. What we have also seen is that it has certain role in ocular diseases as well. But what this study has is, for the first time, we have been able to quantify autophagy markers and show an intracellular signaling mechanism for keratoconus apart from inflammation, which may be in fact contributing to inflammation, and we are trying to develop specific drug targets, and we are trying to extrapolate this in vivo studies into the larger sample size. Now, future directions of this is we are trying to detect this inflammatory as well as autophagy markers and see what we can do further. This is not the end. This was the presentation last year, but this is not the end. This is the beginning. Next. So this is what our research has been doing. What did we do by taking it further? A spark of hope. We had a lot of limitations, lot of questions while we were doing this study for the past 4 years. So right now what we have done is not only detected few of these inflammatory tests, but also the next. We are developing a mechanism. It's a 10-minute presentation. Next. We are detecting these biomarkers on the simple Schirmer strip. Now this will be available to the clinics pretty soon, and all these biomarkers, be it inflammatory as well as autophagy markers, they can be given as a readout on your laptop. So this is the kit that is going to be developed pretty soon and going to be available in the clinics. Furthermore, we have developed one more drug. We are looking into trehalose, which is a disaccharide, which is also an inducer of autophagy. So currently, chloroquine and trehalose are two different mechanisms by which optimal autophagy is derived. I would like to thank these brilliant scientists and to clinician scientists who have been working tirelessly for the thought and the execution of this. Also would like to acknowledge Grow Lab and Narayan Nethralaya staff, and most importantly, my mentor, who whenever one of us or any one of us dropped, he was always there to pick us up, Professor Rohit Shetty. A heartfelt thank and gratitude for him for teaching us and making us much more able than what we are today. Thank you.